Hello, and welcome to Digestive System 1. This podcast, we will deal with digestive system functions. So, if you look at the cartoon on the right hand side, you can see the mouth, also called the oral cavity, that has a mechanical digestion function. So, when you mechanically digest something, you're not breaking chemical bonds, you're just taking something and mechanically making it into something smaller without, like I said, breaking chemical bonds, which would be chemical digestion. So the mouth has a mechanical digestion function. When you chew things, you are breaking the food into smaller parts, and you're mixing that food with saliva. Within the saliva, there's a lot of things, but specifically there are enzymes in saliva that will help begin the digestion for carbohydrates and lipids. And then the mouth also has a chemical digestion function, because if you have an enzyme in the mouth or the oral cavity, that means that enzyme is actually beginning to break down chemical bonds in the food. So the ultimate goal is to take a large food molecule like a protein or a carbohydrate or fat and to break it down into really small parts, or into monomers, in other words, so that those monomers or those smaller parts can be absorbed across the cells of the small intestine. The mouth also has a motility function, so it begins the process of swallowing, and the medical term for swallowing is deglutition. The esophagus, that next part of the tube, so you go from the oral cavity into the pharynx and then to the esophagus. The esophagus has a peristaltic, a motility function, and you move the contents of what you just ate through the esophagus via a process called peristalsis. Peristalsis is just when the smooth muscle cells alternately contract and relax to help push what you now call a bolus of food from the oral cavity through the esophagus and into the stomach. And then when you if you're that bolus of food, then when you get to the stomach, the first thing you'll meet as you enter the stomach is a sphincter. And then the last thing you'll see when you leave the stomach is also another sphincter. So when you're in the stomach, you are susceptible to mechanical digestion function. So because the stomach has 3 smooth muscle cell layers, the stomach is able to churn the food and further mechanically digest the food, or the bolus, I guess you would call it at that point. The cells of the stomach will secrete acids and enzymes. So if you're secreting enzymes again, then you are going to be doing chemical digestion of the bolus in the stomach because you're breaking chemical bonds. Once you've been mechanically and to some extent chemically digested in the stomach, then you move through that distal stomach sphincter into the duodenum. So the duodenum will receive both the stomach contents and the bile that was made in the liver and stored in the gallbladder. The small intestine, that first part of the small intestine, the duodenum, will also receive pancreatic juice, and a main component of pancreatic juice that we've studied in the past is bicarbonate ion. So then as you travel from the duodenum, you go into the jejunum and then the ileum, and in that part of the small intestine you'll complete chemical digestion of all the molecules that you initially ate into monomers, these monomeric forms of these really small parts, so that you can absorb those monomers across the GI tract wall and ultimately to get into your blood. From the small intestine, you go into the large intestine with its subsequent colon parts. The main thing that is absorbed from the colon is water. So you have a water absorption function. So you absorb water back into your capillaries. And then also the colon has a defecation function. The accessory organs then are not part of the GI tract. It's also called the alimentary canal, but they are organs that are vital to digesting the things that you eat into absorbable parts. So the liver makes bile. Bile, this is a tricky question. It could be a tricky question on the test because bile has different bile acids, so you might think that bile is a chemical digester, but it's actually bile has a mechanical digestion function, and we'll look at bile later on and show how how bile does do a mechanical job of digesting. The gallbladder, like I said before, stores bile and releases bile when it's told to do so by hormones. The pancreas, like I said, secretes basic solutions, so secretes bicarb. And the pancreas also secretes a lot of pancreatic enzymes to help chemically digest the bolus that's actually now called chyme. At the end of the stomach, the bolus that went into the stomach, that bolus was processed, and then as it comes out of the stomach and the distal end of the stomach and into the small intestine, it's called chyme. So when that chyme goes into the small intestine, that will trigger the small intestine to secrete a lot of different enzymes to help chemically digest the chyme into smaller and smaller parts so that ultimately they'll be absorbed across the small intestine wall. So here's a picture of a hamburger to show you the different types of foodstuffs that we eat. So carbohydrates are made up of polysaccharides or sugars. Examples of polysaccharides are starch, glycogen, and cellulose. Polysaccharides are chemically digested into disaccharides. Examples of disaccharides are maltose, lactose, and sucrose. Probably know sucrose, that's our table sugar. And then further enzymatic digestion of carbohydrates takes those disaccharides into monosaccharides. So examples are glucose, galactose, and fructose. So those are simple sugars, and those forms, the monosaccharide form, can be absorbed across the small intestine wall. As for lipids, primarily you have triglycerides, so you'll ingest triglycerides, and then those triglycerides can be broken down into smaller parts, glycerol and fatty acids. And then of course proteins can be broken down into polypeptides, and those polypeptides can be broken down into tripeptides that can be broken down into dipeptides. And then when the dipeptides are broken down, you've got single amino acids, and now those single amino acids can travel across the small intestine wall. And then also in this cartoon of the hamburger, or this photograph of the hamburger, you have a nucleic acid because it's beef, and they have, it's muscle that you're eating, this muscle and fat, and those cells of muscle and fat cells have DNA, so they have nucleic acids. And DNA, so that DNA and RNA that you're eating from either meat or plants, DNA and RNA need to be broken down into nucleotides so that they can be absorbed too. So all these processes of chemical digestion, of breaking these chemical bonds of the foods that we eat, are done by enzymes. So this is a quick enzyme review. The blue structure is the enzyme, and that little red structure is the substrate. So enzymes are biological catalysts. That means they speed up reactions. Enzymes are very specific for a certain substrate, so they'll bind one substrate and not another substrate. Enzymes are proteins. That means you need a gene to be transcribed into messenger RNA, and then that messenger RNA needs to be translated into a protein, and then that's your enzyme. Enzymes are very sensitive to the pH that they're working in, the salt concentration they're working in, the temperature that they're working at. And this just shows you a typical reaction sequence. So S stands for substrate, E is for enzyme, so the substrate binds the enzyme, you form a substrate enzyme complex, and then that substrate enzyme complex releases the enzyme plus the product. So the enzyme basically took the substrate and reformed it into a product. Enzymes are reusable tools, so they can be used over and over again. Usually they're doing, in digestion, you're doing a hydrolysis reaction. So you're using water to break the bonds of those those carbohydrates and proteins. So you're breaking bonds to get those large polymers into monomers. Structure of the enzyme allows for function of the enzyme. So if your enzyme finds itself in a pH that it doesn't like, or a salt concentration it doesn't like, or a temperature it doesn't like, it's a protein, so it can denature, and now the enzyme can't work. I have a cartoon of sucrose digestion using the enzyme sucrase, but we'll look at that in class.